Nowadays, one of the biggest challenges we face globally is climate change. Surprisingly, one of the hidden culprits is methane, a greenhouse gas produced in great proportions by livestock and whose warming effects are 30 times higher than CO2. But what if I told you that there was a natural solution growing in the ocean?
The National Hatchery Network is a key development within Australia's seaweed industry. The idea behind the network is to develop a series of hatcheries across the country that will help fast-track the industry. We are specifically focusing on Asparagopsis taxiformis and Asparagopsis armata due to the fact that these species have been recognized and proven to reduce methane within cattle. The project is a collaboration between the Australian Sustainable Seaweed Alliance, James Cook University and the South Australian Research and Development Institute.
The National Hatchery Network for the Culture of Asparagopsis is funded by an $8.1 million grant awarded by the federal government to the Australian Sustainable Seaweed Alliance in collaboration with the Department of Agriculture, Fisheries and Forestry, as well as the Fisheries Research Development Corporation. The main aim is to develop an Asparagopsis industry in Australia, which can help to reduce the methane emissions of livestock.
So the way that Asparagopsis is able to mitigate methane emissions and contribute to climate change mitigation is because it's been shown many times now through many studies that when this species is included at very low doses in the diet of ruminant livestock, mostly from cows, that it can dramatically reduce the production of methane by consistently around 90%. This is really important because methane emissions from this source contribute to about 18% of global greenhouse gas emissions, and this methane that's being produced is a lot more potent than carbon dioxide, which we often all hear about. Half of those emissions from that 18% are coming from this digestive process. The way that Asparagopsis is able to have such a potent effect is because it produces a specialized compound called bromoform. And that's a bioactive substance that's produced by the seaweed itself, and it acts by blocking one of the key enzymes involved in the production of methane during that digestive process. The National Hatchery Network is committed to contributing to this climate mitigation and reducing enteric methane emissions through developing sustainable seaweed aquaculture. Primarily, we're focusing on Asparagopsis, but we're also looking to expand into other seaweeds as well in order to really develop this industry.
Asparagopsis has a complicated life cycle with 3 different life stages, making it challenging to close the life cycle under aquaculture production. Achieving a high degree of control over life cycle transitions is important for effective management of any farm species, and is also important for future selective breeding of desirable traits, such as high bromoform content, fast growth, and also enhanced reproductive output. Contamination also poses a major risk for seaweed production, as it can reduce the overall productivity of the targeted species and also impact the bromoform content of the final product. There's also a risk of contamination overtaking the culture, which can lead to substantial biomass loss and also the need to restart the culture again from scratch.
One of the key components of the National Hatchery Network is the development of the Tropical Hub. The Tropical Hub will focus on research around Asparagopsis taxiformis and has a number of key objectives. The first one is to establish facilities and capacity for this research. The second is to develop a research program for closing the life cycle, specifically of Asparagopsis taxiformis. The third is to develop decontamination techniques for long-term maintenance and storage of cultures. The fourth is to develop and improve on current technology for the production of the species. And the final objective is to contribute towards a manual that can be used by first-time farmers. Here at JCU, we have dedicated facilities for the NHN, which include dedicated laboratories, both wet and dry labs, office space, as well as the production equipment for upscaling cultures. We have developed a number of clean stocks that we've collected on field trips from locations around Queensland, and we have kept both wild stocks and produced our own clean cultures with these as well. Within the equipment that we have here at JCU, we have developed industry-relevant techniques, specifically the use of bag cultures adopted from microalgal production. And these are being used to upscale production in a sterile and efficient manner. Furthermore, we have the potential to move to even larger cultures using our dedicated raceways within our greenhouse.
One of the key challenges in upscaling Asparagopsis cultivation is managing contamination. Contamination is inevitable when the seaweed is collected from natural environments. Our original samples are collected from the wild, from both on shorelines and from fringing reefs. Our sampling methods in the wild involve collecting live material and then the preservation of that material for transfer into our labs. Because environmental conditions can vary a lot, so can the degree of contamination in our wild collected samples. We're testing various treatments, including biological, chemical, and physical, to reduce and eliminate contamination in wild-collected Asparagopsis cultures. We found that it's possible to significantly reduce contamination while still maintaining positive growth rates by carefully manipulating several factors. So far, the only consistently reliable method involves isolating tiny, clean fragments of the tetrasporophyte and then growing that up into larger cultures. It's also possible to use the spores released from reproductive gametophytes to create clean tetrasporophyte cultures.
Reproductive events in seaweeds are primarily triggered by different environmental factors, and these are often related to seasonal change. So things like temperature, photoperiod, light intensity, and nutrients can all induce these reproductive transitions. And what we're trying to do with Asparagopsis at the JCU National Hatchery Network is develop a comprehensive research program to close these different stages of the life cycle that would naturally be occurring in the wild inside the lab. And that's because we want to be able to obtain high quality spores that we can use for setting onto ropes and optimizing production in that sense. Over the past few months, we've been working on developing this and have successfully induced the production of spores across several different strains, which is really exciting. But now our focus is more shifted into understanding how these different environmental factors, specifically temperature, vary between these different strains. The outcome of this is to provide farmers with a framework that they can use to inform how they can optimize the production of those spores across different strains depending on where they've got their biomass from. We're also conducting similar environmental manipulations experiments using these different environmental factors to try and optimize not just growth but also the production of that bromoform, because these factors can also influence both the growth and the production of those bioactive substances.
Having set up two hatcheries, one in the tropical location and one in the temperate location, allows us to build up clean seed stock in the respective hatcheries, and this will be made available to farmers, allowing them to reduce their own startup costs as well as to upscale their farm production much faster than would be possible otherwise.
The NHN has 5 major key outcomes. The first one is obviously to increase the production of Asparagopsis within Australia, but also hopefully globally as well. And this will be achieved through the improved understanding of the species, but also the development of new hatchery techniques, which will address key bottlenecks such as increasing spore production, the reduction of contamination, and upscaling culture biomass. Through all of this and the increased production of Asparagopsis, we hope to significantly contribute to the reduction of greenhouse gas emissions through the use of Asparagopsis as feed within the cattle industry.
The significance of the National Hatchery Network is that it will help lay the foundation to fast-track the development of a sustainable seaweed industry in Australia. This industry has the potential to grow to a value of $1.5 billion by 2040, providing 9,000 jobs, mostly in regional and rural Australia. The major output of the National Hatchery Network is the production of a hatchery manual, which will be made available to the public. This was identified by stakeholders as one of the major obstacles to the development of a seaweed industry, namely the lack of expertise and publicly available knowledge.
In conclusion, the NHN will not only fast-track the increased production of Asparagopsis within Australia, but also help pave the way to combating climate change. I think it's also really important to realize that although Asparagopsis is currently the poster child for the NHN, there are a number of other species that hold significant benefit for the development of this new and emerging industry. Here at JCU, we are currently investigating a number of novel uses for seaweed within both aquaculture and environmental improvement. I think that the fact that 70% of the globe is covered with ocean and that seaweeds are really the crop of the sea, we really need to understand how these important resources will contribute towards future food security.
Thank you.